401 General Biochemistry. As we always do, your microphones are currently disabled, but the chat is available. I'm monitoring it. So if you write in the chat, I'll see it. If you have any concerns, if you have any questions, any contributions, you can write in the chat and I'll see it. Alternatively, you can jot down your comments, your questions. At the end of the lecture, I will open the floor so you can raise your hand and I will enable your microphone. Call your name, you speak, and I respond. We previously finished with, I think the last time we did something on enzyme. I think we finished with classification, naming, and the last lecture we did mechanisms of action of the enzymes. So today, I think today should be the last class on the enzymes. We'll finish, that, first part of the course, which is on the enzymes. So we'll finish with some enzyme kinetics. So what we'll do today is to take a look at some, basics of enzyme kinetics, and then we'll continue to look at some practical applications. We'll select a few of practical applications of enzyme inhibition, and then we'll end it there. Of course, this is a lecture that we cannot cover all the things you can talk about with enzyme inhibition and its applications, particularly in the pharmaceutical industry, in the discovery of drugs, or even in the use of pesticides. It's a whole lot that we just cannot say that over a period of an hour or two you can cover these things. So hopefully, as we always do, we spend about an hour, a little bit more, listening to me, and then after that, the floor is open for you so that I can also listen and respond. So we can start. First of all, to understand what enzyme kinetics is. You remember we studied the factors that influence enzyme activity. That was in the previous lecture, where we looked at factors such as enzyme concentration, substrate concentration, temperature, pH, as major factors that influence enzyme activity. Now, kinetic studies on enzymes is basically studying the rate of reactions. That is a way to measure the activity of the enzymes depending on changes of these experimental parameters that we studied earlier. That is the pressure, the pH, the temperature, substrate concentration, and enzyme concentration. So what you do is you keep, say, other factors constant, and you vary one of the factors. Like for instance, you vary pH, or you vary temperature, or you vary substrate concentration, or you vary enzyme concentration to see the response to these changes. So that is how you can determine how the enzyme activity is influenced by these parameters. So when you do that, what you are doing is what you refer to as enzyme kinetics, or you're just doing some kinetic studies on enzymes. Now, with that introduction, let's take a brief historical background to the simplest form of representation of enzyme kinetics, which has been accepted and is used all over for a very long time now internationally. This has been the accepted representation of enzyme kinetics, what we refer to as the Michaelis-Menten kinetics. And we come to see the Michaelis-Menten equation. All right, so let me see. Somebody is writing. He says, this is Salin John. He says, this Aya Salin John, please. I'm using, but I can see your name there. All right, so was first reported by Victor Henry. The first picture you see on the left. Victor Henry is actually a Russian, but he was born in France. So on paper, is French, but he's actually Russian because his parents are Russian. Well, the situation in which he had to become a French instead of a Russian, I will leave that. If you are interested, you can go and read more about it. Why he had to become a French instead of being a Russian on paper. That's another story altogether. That is away from the biochemistry. It's a family issue. You can read on it. He was the first who actually represented the relationship between substrate concentration and enzyme activity. I mean, in his but it was not fully understood. It was about some 4 decades later that is like 40 years later that Leonard Michaelis, who was a German chemist and physician at the time, he was also doing some enzyme kinetic studies. Actually, he was German, but he later had to travel to Japan. He worked in Japan for some time, and later he had to go to the United States, and that's where he later retired. So he retired in the United. He wrote a paper that got him into a little trouble, and he felt that he would not be, having his promotions and, enjoy his benefits. So he had to leave and go and spend some time in Japan. Well, now Leonard also into these enzyme kinetic studies actually worked with, the last lady on the right, that is Maud Leonora Benton. Now, this time, I want the ladies to listen carefully to the story of Leonora, because I think it is that inspirational that at the time, she was actually one of the first female medical doctors in Canada. She's a Canadian. And after her medical studies, she did a bachelor's and did a medical studies. She was interested in going into scientific research. But at the time, women were not allowed to participate in scientific research. So Aful is saying some people are struggling to join the class. I think nobody should have any issue joining the class. You joined easily. There was no problem. Everybody else joined. So if anybody is having any struggle, it is obviously coming from they themselves. They should deal with it. So you have Leonora wanting to participate in scientific research. She wants to go into scientific research, and yet at the time in Canada, it was impossible for women to engage. She worked together with Leonard Michaelis. So that was when they came up with the Michaelis-Menten equation. They were actually, building up on the work of Victor Henry. So sometimes it's said that instead of referring to the Michaelis-Menten kinetics, if you want to, historically be accurate, you should rather be saying Henry Michaelis-Menten kinetics or Henry Michael-Menten equation. When we come to that, you see the equation that I'm talking about. So Leonora and Michaelis, represented more accurately how substrate concentration influences enzyme activity. Now, later, she also continued, left Germany, went to the United States, and continued there, became a professor. And she actually became a professor in the last year of work, like, in active service. At the time, they retired at the age of 70, and she was 69 before, and she got her PhD. She became a professor at that time, just the last year when she was retiring. If you were someone, some people would just say, after all, I'm going to retire, so there is no point, pursuing anything again. But that is a woman that was so, hardworking and wanted to get to the top. So this is why I said, sometimes I think that apart from the technical things that we learn in these things, we should listen to some of the inspirational stories behind some of these things so that we don't easily just throw our hands up when we face difficulties and stuff like that. So, that's a bit of a historical background of the Michaelis-Menten kinetics. Now, what is this Michaelis-Menten kinetics about? So, as I was saying from the beginning with the introduction, that you vary experimental parameters and see how these parameters influence the activity of enzymes. So you take a look at these enzyme kinetic studies, which is represented on this graph here. We have on the vertical axis substrate concentration, and on the horizontal axis we have time, so reaction time, the time over which the reaction was occurring. And the observation by Michaelis and Leonora was that, if you take a look, you see that substrate concentration was high from the beginning. And you have the red line is for product concentration. And the product concentration is 0 from the beginning. So, there was no product yet. That's the beginning of a reaction. and the substrate was there. You know, those times usually the enzyme they used for studying these things was invertase, which was responsible for the breakdown of sucrose, the table sugar, into glucose and fructose. It was the same type of studies that Henry Victor also did. And a lot of times, in those times, that was the enzyme which was being used in these enzyme kinetic studies. So we take it that the substrate was sugar, the table sugar. And then you have enzyme, which is also represented here with the blue dash lines. So the enzyme being invertase, responsible for breaking down that sugar into the monomers, glucose and fructose. And you have a brown or dash line also representing the enzyme substrate complex. So we've seen these things already, so you can quickly relate to them. So it's clear that at the beginning of the reaction, you have the substrate being high in concentration, product 0, no product yet. Enzyme, you also have enzyme concentration of a certain level in the solution. And you have, of course, from the beginning, no enzyme substrate complex yet formed. It is when you have the substrates now binding with the enzyme, then you begin to have the enzyme substrate complex being formed. And you see this, sort of influence that you have the enzyme substrate complex increases up and then goes down, and you have the enzyme concentration also, sort of reduces because it's now binding to the substrates and forming the enzyme substrate complex. So it reduces all the way. We'll study we'll look much more into detail of each of these stages that are looking this way. And you have the product, of course, once you have the enzyme substrate complex formed, you begin to have products that will be formed from the transition state. And you have the product concentration increase all the way till the maximum, and then you have substrates reduced all the way to 0 when you have all the substrates consumed in the reaction mixture. All right. Now, if you take a look at this behavior of the enzymatic reaction which we were looking at, you realize that there are different stages occurring. It is actually these, specific stages that Henry Victor did not particularly focus on. He just looked at the, reaction proceeding over a period of time, looking at substrate concentration and just, developed an equation out of that. But it didn't take into consideration, all of these, stages that we are looking at. So for instance, you have the green shade part where you are having the enzyme concentration up there, and you have the enzyme substrate complex concentration low there, where you have these concentrations being wide apart. It is actually in this region, this green region. So within this period of time, you are having the formation of the enzyme substrate complex. That region was referred to as the pre-steady state. So the reaction was not moving steadily yet. Then you come to the brown region, where you can see that when we were looking at the influence of substrate concentration curve, where we said that you reach that saturation of the enzyme such that any amount of substrate you are adding will not be resulting into any change. And so you have a steady state. That is a steady state. The rate of the reaction is going to be constant. So if you take a look at this blue curve for the enzyme and the brown curve for the enzyme substrate complex, you realize that in this brown region, these two curves are relatively steady. They are relatively constant. So they are looking almost just like a straight line there. It is actually in this region that you are having the enzyme substrate complex concentration being relatively constant. So you have enzyme substrate complex concentration relatively constant, and you are also having the enzyme concentration being relatively constant. Of course, at this point, you are having enzyme molecules, bound to the substrate to form the enzyme substrate complex. So you see that the concentration of the enzyme substrate complex is gone up, whilst the concentration of the enzyme is gone down. Now, once products begin to form and the concentration of the substrate also continues to reduce, you see now that you are having more and more product formed until the maximum concentration of the product is reached, whilst you have the substrate fully consumed out of that reaction mixture. In that same manner, once you have more of the enzyme substrate complex going into the formation of products, because that enzyme substrate complex, will go to the transition state and then form products. Then it means the enzymes will be getting free until the concentration of the enzymes is what you get at the end. So the enzyme concentration is now the same as it was from the beginning. And you also see that there will no longer be any enzyme substrate complex there when you have, all the enzyme substrate complex now converted into product. So that was more of a consideration of the mechanism of reaction that will occur in an enzymatic reaction, looking at all of these stages. So Michaelis and Menten were able to come up with, a simple mathematical equation to represent the influence of substrate concentration on enzymatic reactions. And this is a curve that we saw much earlier. Now, if you look much more onto this curve, we have what we refer to as the Michaelis-Menten constant, which is represented Km. Now, Km is the substrate concentration at which half of the maximum of the rate of reaction is reached. So remember, we said the maximum rate of the reaction, which is shown with the dashed lines at the top there, that's the possible maximum rate of the reaction that can be reached. Above that, you cannot have anything else occurring because you have all the enzymes being occupied in that reaction mixture. So if you have a maximum rate of a reaction there, to reach half of that maximum rate of reaction, you must have a certain concentration of substrate in solution. And that concentration of substrate is what is referred to as the Michaelis Menten constant. And so this is a reaction which is used in enzyme kinetic studies. There are some others like the Lineweaver-Berg, Eadie-Hofstee, things like that. But those other representations actually have some elements of error such that it is not recommended to be using those reactions to those equations to be calculating parameters in enzyme kinetic studies. And so it's better just to stick to the Michaelis Menten kinetics, which is used worldwide to study enzyme kinetics. And the equation, is based on, if you remember the equation of a line where you have y is equal to ax plus b, that is here, it means that y is going to be the rate of the reaction, and you have the x being the substrate concentration. Now, A is going to be the what you call the gradient. These things you did them secondary school, so of a straight line. So you take, say, the linear range from, say, 0 going up. If you draw a tangent, a straight line up there for that estimate, obviously the gradient is going to be taken from where you are seeing the half maximum of the reaction and the Michaelis Menten constant. And as from such equation of a line, you could derive the equation of the behavior of an enzymatic reaction with influence of the substrate concentration, such that you have the rate of the reaction being equal to Vmax, that is the maximum rate of the reaction, and a product of it with the substrate concentration divided by the Michaelis Menten constant, which is what you see on the graph, plus enzyme plus substrate concentration. So you can actually derive this experimentally. What I mean is that if you do an enzymatic kinetic studies, such that you have different concentrations of a substrate. Imagine you have some sugar solution, you have different concentrations, and you have the enzyme, say an invertase, or let's say you have is it a galactosidase? Galactosidase will be acting on lactose. So let's say some hexokinase or glucokinase that is acting, you find some color changing reaction, so that you can use maybe spectrophotometer to measure, the intensity of the color, which is going to be, corresponding to the rate of the reaction. And you have a graph of a sort, you can easily determine the Michaelis Menten constant. And these substrate concentrations are also known. So at any, and you have these absorbance values there. So which means in place of the rate of reaction, what you are going to have will be absorbance values. So you can easily determine just by, plotting the chart, absorbance against substrate concentration, you can easily determine the Michaelis Menten constant at any point in time. Now, these days, because you have a computer that you can use to quickly, plot these graphs, which is much more the reason why it is not advisable to be going for the use of the line weaver bed plot, for instance, that you have to now be converting this graph into a linear graph and things like that. So we'll not go into those types of representations of the kinetics of enzymes. Previously, we did with the other years, but I think we should rather focus on just what is, being used, especially in this computer age, and then go on and look at uh, some practical examples of what we are doing and all of that, instead of going to be looking at those things which are not being used. There will be no need doing that. All right. So we move on to what we refer to as enzyme inhibition. I think we talked briefly about inhibitors, which are compounds or molecules that can slow down chemical reaction or terminate the chemical reaction. So we'll look at some forms of enzyme inhibition, and then we'll look at the application of enzyme inhibition as pharmaceutical agent in the pharmaceutical industry, as pharmaceutical agents, the use of these inhibitors. Enzyme inhibition can also be used to study, enzyme reaction mechanisms. I mean, that is to sometimes study what will be the activation center. If you know a particular inhibitor molecule that you are using, what can it bind to, or just to slow down a reaction to be able to study it more steadily. If you are able to introduce some enzyme inhibitors into reaction mixtures, obviously, when the reaction is running slow, you have enough time to even go ahead to study at various intervals some intermediaries that are formed in a reaction. I mean, using methods like say infrared spectroscopy, the nuclear magnetic resonance spectroscopy. You can use these methods to actually determine the chemical structures of the compounds that are in a mixture. That's going to let you know that at any point in time, you have some intermediaries of this structure, things like that. Of course, those are time-consuming, complex studies to do that can take you months, if not years, to come up with the various intermediaries and propose a reaction mechanism that's a particular enzymatic reaction pathway is following. All right. Now we take some forms of inhibition. The first we're looking at is competitive inhibition. And as the word suggests, I think we saw some competitive inhibition much earlier. We mentioned it briefly. So this is an enzyme, red. The enzyme is shown with a red color, and the yellow object is depicting the normal substrate that should bind into the active center of that enzyme. But you can have another molecule which looks like that normal substrate. That becomes a competitive inhibitor because it will be competing with the normal substrates for the active center. So when it binds in the active center, it prevents the substrate from coming to bind in the active center. So the reaction is inhibited. The reaction is either terminated or it is slowed down because it will take more time for that inhibitor to be displaced. That is if substrate concentration is increased for the inhibitor to be displaced from the active center. So that is competitive inhibition. Now, competitive inhibition can be represented by the Michaelis-Menten plot, such that if you take these charts that we are looking at, it's just like the chart we looked at much earlier, where we are seeing the rate of reaction on the vertical axis against substrate concentration on the horizontal axis. The red curve represents an uninhibited reaction. That's an uninhibited reaction. And the blue curve represents an inhibited reaction, a competitively inhibited reaction. Now, what you notice is that Michaelis-Menten constant, which is the substrate concentration at which half of the maximum of the rate of reaction is reached, that has increased for the inhibited reaction, which is the blue curve. And this is logical. I mean, you are having some amount of enzymes in a reaction mixture. Then there are inhibitors that have bound to the enzyme molecules. It means that you will need more substrates in the reaction mixture in order for you to even reach the maximum rate of the reaction. That's first of all, to reach the maximum rate of the reaction. You will need more substrate, because first you need substrate that will go in there to displace the inhibitor from the active center before you have the reaction proceeding. That also means that you will need more substrates, molecules, Michaelis-Menten constant, which is higher than the Michaelis-Menten constant for the uninhibited reaction. Of course, in this case, you can still reach the maximum rate of reaction. The maximum rate of reaction has not changed. It hasn't changed. It is still the same. So that's for competitive inhibition. So if you practically, you are running enzymatic reactions in the laboratory, what it means is that if there are some inhibitors in there and the reaction is slow, you are not getting, the amount of products that you should get. Once you add more substrate in there, those substrates will, because it's a competition. So once you have more substrate, you increase the probability of the substrates binding to the active site. And at the same time, even when you have some inhibitors already bound, the substrate molecules, because they are high in concentration, can go in and displace these inhibitor molecules and have the reaction proceed. So that is a representation of the competitive inhibition by the Michaelis-Menten plot. Then we have the non-competitive inhibition. So here we see clearly that apart from the active center where the substrate is binding, the enzyme also has, another site, usually referred to as some allosteric site, to which you have a non-competitive to another site, not the active center. And once it binds to that, substrate, enzyme substrate complex, it renders the enzyme substrate complex inactive. It cannot proceed to the formation of a product. So that is the non-competitive inhibition. Now, by the Michaelis-Menten plot, you can also represent the red line is representing the uninhibited reaction, and the blue curve is representing the non-competitively inhibited reaction. Now, what you see is that the Michaelis-Menten constant is the same. That has not changed. In competitive inhibition, the Michaelis-Menten constant changed. But here, you see the Michaelis-Menten constant is the same. It means that it is at the same substrate concentration that you achieve half of the maximum rate of the reaction in both cases. Except that you see, of course, you have the maximum rate of the reaction being different. And so you have, different because, of course, you have the maximum rate of the reaction reduced due to the fact that you have an inhibitor in that reaction mixture. Now, of course, you see the maximum rate of the reaction is changed because the inhibitor doesn't have to compete with the substrate. So whether there is substrate there, increase the rate or not, the inhibitor has its own sites that it will bind to, and so it will reduce the rate of the reaction, the maximum rate of the reaction. You will not be able to reach that maximum rate of the reaction, which means half of the rate of the reaction too is going to reduce. But the substrate concentration at which you reach both of these half of the rate of reactions is constant, is the same in these two situations. All right. Now we can also look at inhibition in two ways, that you have some reversible and some irreversible inhibition. So reversible means that you have inhibitors that bind to an enzyme, but those inhibitors can dissociate, from the enzyme. After some time, they can dissociate. Like what I was talking about, the competitive inhibition, when you introduce substrate, the substrate can go and displace. That is in the case when you have reversible inhibitors. Irreversible inhibitors. So Williams-Ankara says, please, is it a graph of rate of reaction, that is V, to substrate concentration? Exactly. Williams-Ankara, that's what it is. So you have some reversible, you have some irreversible inhibitors, which is when they bind to the enzyme, they bind with some covalent bonding or some, very strong bonds that are permanent. They no longer, get dissociated, and so there is permanent damage done. You see some pharmaceutical agents, some drugs that are such irreversible inhibitors. You also see some of these which are reversible inhibitors. So you can have inhibitors of these two kinds, reversible and irreversible inhibitors. Right. Now we can begin to look at some very practical applications of this in reality. Let's take some real examples and look at. Of course, we can't cover all of. There's a lot of things to. It's not possible to deal with all of these things, but we can take some examples, and then perhaps our appetites will be whet so that we go and do more research. Sometimes if you just go to the pharmacy and you buy medicine and you take the leaflet out, you start to read it and you see mechanism of action of the medicine, you begin to understand, this medicine acts on a certain enzyme, and then you quickly go and check. So what is this enzyme? What does it do? So how will the medicine, inhibit the enzyme? And when the medicine inhibits the enzyme, what will be the result? I mean, that's how these, studies should, make you that if you go and you buy anything, like if you buy something, you read, then quickly you are beginning to understand that this medicine acts in this way. Or you go and buy some pesticide, some insecticide, for instance, and in the insecticide, quickly you start checking. So this active ingredient, what does it do? It's an inhibitor to an enzyme. What does the enzyme do? Okay, so this active ingredient actually inhibits this enzyme. And when it inhibits this enzyme, this is the effect that it causes in the insect, things like that. So that is the basis that this meeting should give you. And then once your appetite is whet, you can go and be doing research. And when you see anything, you are conscious and you are looking at it more and more into detail. So let's start with the first story. So we start with the story. So we have the story of the biosynthesis of tetrahydrofolate. And tetrahydrofolate we saw before. We saw tetrahydrofolate as one of the coenzymes, one of the vitamin Bs, what we refer to commonly as folic acid. You remember very well. So we have tetrahydrofolate that is synthesized in the body. So you have biosynthesis of tetrahydrofolate. And how is tetrahydrofolate synthesized? So tetrahydrofolate is synthesized, represented here in three simple reactions. See, the arrows are three simple reactions where you have a dihydroterine pyrophosphate and 4-aminobenzoic acid. I mean, from the top, I'm reading from the top, the red molecule with two phosphate groups on it. It's a pyrophosphate or a diphosphate. And quickly you can relate to it as say, I mentioned that lots of these, the molecules get phosphorylated. Is to activate them to go into a reaction. So you'll be seeing them you'll be seeing these phosphorylation reactions over and over again. And you see 4-aminobenzoic acid, which is blue, which sometimes is also referred to as para-aminobenzoic acid. Para means opposite. So if you take the six-sided ring, you see the amine group, is directly opposite the carboxylic acid group. And if you are also taking this carbon on which the carboxylic acid group is as carbon number 1, and you count that carbon number 1, then you count 2, 3, 4. It is on the 4th carbon that you find the amine group. That is why it is also written as 4-amino benzoic acid. So it's either you say 4-amino benzoic acid or para-amino benzoic acid. It's the same thing you are referring to. Now, you see, the first thing which occurs is that you have the hydroterine pyrophosphate and 4-amino benzoic acid getting conjugated. I mean, this is very conducive. You can clearly see the red portion and the blue portion that are bonded together. And so you have that amine group that is, losing one hydrogen there. The phosphates, those 2 phosphate groups are gone with the oxygen, and you have a dihydroteric acid as the first, intermediates that you have there. The reaction continues, and now you have the conjugation of glutamic acid, which is green. The glutamic acid residue is green at the end. So you see here, the blue side is that is the end, but now you see that there is a conjugation of glutamic acid in that reaction. Then the next thing, and so that now gives you a dihydrofolic acid. You now have a dihydrofolic acid. Now, the next reaction that you see is that you are having some form of reduction of that molecule. Now, let's pay attention. What is the difference between this dihydrofolic acid and the last molecule? That is the end product, which is the tetrahydrofolic acid. What's the difference between these two? You have to pay attention to see that. You see, in the dihydrofolic acid, look at this nitrogen there. This nitrogen does not have a hydrogen on it, and it's even having a double bond there. If you come down to tetrahydrofolic acid, you see that nitrogen now has a hydrogen on it, and the double bond is gone. So that's a form of reduction. There is some transfer of a hydride group onto that nitrogen, and the double bond is also gone. And so that reaction is a reduction reaction. That's a reduction reaction. That's why you see the name of the enzyme which is involved in that reaction is dihydrofolate reductase. So that is a reduction reaction. Any other thing you look at is the same. So if you look closely, not at all parts of the molecule compared to that dihydrofolic acid up there, everything else is the same. So it is only on that nitrogen that you have that, reduction reaction, which is catalyzed by dihydrofolate reductase. Now pay attention to that. The first reaction up there is also being catalyzed by an enzyme, dihydroterate synthetase. That is the first enzyme. So we are talking about 2 enzymes here in this reaction pathway. The first enzyme in the first reaction, dihydroterate synthetase. That's the first enzyme. And then in the last reaction, that is the third reaction, you have a second enzyme, which is dihydrofolate reductase in this reaction pathway. All right. Now, this biosynthesis of tetrahydrofolate understood. And you also want to remember that tetrahydrofolate, or what we refer to as folic acid, was involved in cell proliferation and at different stages of the cell development. So I think we saw that tetrahydrofolic acid was involved in, say, development of the neural tubes, let's say the brain or the central nervous system of the fetus in the uterus. And that's why it's very much required in pregnancy for some women. Now, the same way, tetrahydrofolic acid is also involved in cell proliferation, of bacterial cells, bacterial cell proliferation. So bacteria, the growth of bacterial cells, their reproduction and all of that. Tetrahydrofolic acid is needed there. Folic acid is needed in those different stages of these cells. Great. Now that's understood. So what we'll do is that we will zoom in on the first reaction. So we are looking at only the first reaction. Remember what I say, one arrow at a time. So we have taken just the first arrow, And the reaction is dihydroterine pyrophosphate reacting with the enzyme which is involved in that reaction is dihydroterate synthetase. Now you see that arrow, there is a cross on it, and there is a small note beside the cross, which is sulfonamides. Sulfonamides are a class of antibiotics. Now, what a cross means is that these sulfonamides act as inhibitors to the enzyme dihydroterate synthetase. It means that when these sulfonamides are administered, they go in there and prevent the first reaction of the synthesis of folic acid, which is needed for the bacterial cell growth. So if the bacteria is not able to synthesize these folic acids, obviously it will result to bacterial cell death. Now, you see clearly, enzyme which is being inhibited is dihydroterate synthetase. What will be the substrate to that enzyme? The substrate to the enzyme is para-aminobenzoic acid or 4-aminobenzoic acid. Now, if these sulfonamides are acting as competitive inhibitors, then they should have some structure which looks like the para-aminobenzoic acid. So it's like they come in to deceive the enzyme. The enzyme, they come and bind to the enzyme as if they are the substrate. But because it's not a substrate, the reaction will not proceed. So you see, intentionally, I've brought here the general structure of sulfonamides, which are also sometimes referred to as sulfanyl amides. And when you look at it side by side with the para-aminobenzoic acid, you see the similarity. You have the six-sided ring with that amine group there. So you have they are attached onto the ring, and For para-aminobenzoic acid, you just have the carboxylic acid group there. But in sulfanyl amides, you will now have this group of, the sulfoamide group, which is bonded there in place of the carboxylic acid group. Looks similar, sort of a structural analog that can bind in the active center of the enzyme and prevent the substrates, the original substrate, which is the para-aminobenzoic acid or 4-aminobenzoic acid, to have interaction with the enzyme dihydroterate synthetase. All right, now let's take the last arrow, because if you pay attention, dihydrofolate reductase. Hydrofolic acid was getting reduced. So you now have a hydrogen on it, and it loses that, that double bond is removed from the molecule. Here you see also that there is a cross on that arrow with a small note beside it. Trimethoprim. Trimethoprim is a drug that serves as an inhibitor to the enzyme dihydrofolate reductase. So when the enzyme is inhibited, this reaction will not proceed. So you don't have the tetrahydrofolic acid, what we are referring to as the folic acid, will not be synthesized at the end because of the drug trimethoprim, which is introduced in there. So you see here too, the enzyme this time around is dihydrofolate reductase. The substrate to that enzyme is the dihydrofolic acid. And you see trimethoprim sort of having some similarity, not so much, not so specific. You see that ring, the two ring structures, they have some separation between them. In place of the double bond O on the terine ring, you are having an NH2. This NH2, this amine group is the same there. The rest of the things are not the same. They are not so much similar like what we saw with the sulfanyl amides. But trimethoprim can also serve as an inhibitor to this enzyme. Now, we are taking the same reaction again, that last reaction. This time around, the inhibitor is methotrexate. This drug, which is, I think we saw it used in chemotherapy against cancers. And I mentioned they go in there and prevent the synthesis of tetrahydrofolic acid, which is required for proliferation of cells. And because you have cancer cells that are proliferating so fast, so if methotrexate goes to inhibit that, sort of brings down, the growth of tumors and can even result into the cancer cell death. And you see methotrexate, if you look at the structure, this is looking much more. So you see how, like, specificity of drugs, the more the drug is looking much like the substrate, the more specific the drug is going to be. Because now if you look at methotrexate, the structure over here, it looks very much like the tetra hydrofolic acid, not the tetrahydrofolic acid, the dihydrofolic acid. The dihydrofolic acid that is converted by the dihydrofolate reductase to the tetrahydrofolic acid. If you look at this structure of methotrexate, it looks very much like it. So let's see, if you look at the first ring, in place of the double bond O on that ring in the dihydrofolic acid, for methotrexate, the drug, you have an NH2. So that's a difference you have there. Where else can you find a difference? On this nitrogen, you have a methyl group, CH3. CH3. But in dihydrofolic acid, there is a hydrogen. Where else? You can see on the two rings for dihydrofolic acid, the rings that are fused together, these two nitrogens are having hydrogen on them. They have hydrogen on them. If you come to the drug, methotrexate, there is no hydrogen on those two nitrogens. And there's even a double bond that has appeared there because the hydrogen is not there. You have double bonds on those nitrogens. I think everything else is the same. If you look at it clearly, everything else is the same, except for those things that we have pointed out now. So you see, there is that, similarity to structure, and so they can act as, competitors. You have methotrexate acting as a competitor to dihydrofolic acid, competing for the conversion of dihydrofolic acid to tetrahydrofolic acid. All right, so that's another example that we have taken a look at. Now let's start another story. This story we have actually seen it before, but maybe we see it in a different way. This is about the component that is the major, part of the cell walls of, gram-positive bacteria, what we refer to as peptidoglycan. I think we saw when we were looking at lysozymes. I think when we were talking about lysozymes, we talked of the peptidoglycan, which is made up of N-acetylglucosamine and N-acetylmuramic acid. Here we have the violet, spheres being N-acetylglucosamine, and we have the blue spheres being the N-acetylmuramic acid, which are bonded together. So you have this heteropolysaccharide, derivative, because you are having this glucose molecule with some, glycosyl group and some muramic acid group that is attached to it. So you see so if actually you look below here, if this is the bacteria and you zoom in on, the outer membrane, you have, the double layer phospholipid, which is the plasma membrane, and then above it, which is the thick black line, is now what is, being zoomed in to show as the peptidoglycan layers, or the peptidoglycan cell wall, which is a protective coating for the bacteria. So you remember that. And you see that there is some kind of a cross linkage between these uh, um, you know, heteropolysaccharide that is made up of the N-acetylglucosamine and N-acetylmuramic acid, which is shown with these red dashed lines between them, what we are referring to as some cross-link, peptides between them. So you have some peptides that link the uh, heteropolysaccharides together. That's why we refer to it as peptidoglycan. The glycan is the carbohydrate part of the structure. And those peptides, those short peptides that are linking these glycans, make it a peptidoglycan because they have some peptide bonds in there that are linking these um, polysaccharides together. All right, so now we have seen that, basic structure of the peptidoglycan. So we can continue to talk about, some enzyme inhibition in other ways. But now we look briefly at how the peptidoglycan is synthesized. So we're looking at the process of biosynthesis of the peptidoglycan. Particularly, we are referring to the formation of the peptide cross-links. That's what we are looking at. So we take this one to, we've numbered it 1 up to 5. So it's from the top down. So we can quickly relate to it. Now, you see the straight, chain of the N-acetylglucosamine, N-acetylmuramic acid. That is the carbohydrates, the glycan part of it, such that, this teal, this teal color glucose glycan chain. Now you can see that on N-acetyl muramic acid, you are having the peptides. So you have some peptides on N-acetylmuramic acid residue on the green portions, all of them all the way down. Pay attention, you see that you have some short peptides on them. And these peptides too, we have shown that these red spheres are alanine. The green is lysine. And you have the blue being glutamic acid or glutamate. So these are the amino acids that are in that peptide chain. Then you see there is a protein we are referring to as penicillin binding protein. In this case, because we are looking at biosynthesis, let's refer to it as glycopeptide transpeptidase, because that's what that is the name of it as an enzyme. That's what it does. But because it is that enzyme which is targeted by the antibiotic penicillin, it is also sometimes, it is also referred to as penicillin binding protein. But as an enzyme, it is glycopeptide transpeptidase. And you also pay attention and see there is something small there inside, violet, and it's written beside there, it's a reactive serine. You remember we looked at one of the mechanisms of action of enzymes, and we called it mechanism of action of serine proteases. So you can see this is a serine protease. It's having a serine residue in the active center, which means the activation of it there will also be in the form of proton transfer. We saw that mechanism. And here you see, what is the glycopeptide transpeptidase doing? It is responsible for the formation of the peptide cross linkages, what we saw in the structure of the peptidoglycan. So you see, when we come to number 2, you see the enzyme glycopeptide transpeptidase comes in and binds onto one of these peptides. And what do you see? You see that that red sphere refers to alanine. That alanine is interacting with the serine in the active center. Come to number 3, and you see that you have a second peptidoglycan that is binding into another side of the active center. And you now have number 4, the cross linkage. And that cross linkage is between alanine, which is red, and green, lysine. And so you now have that cross linkage, which is shown yellow, is also being named as the pentaglycine chain. You are having a chain between lysine, the green, and alanine. So you have this occurring. It means that between the others, the 2 here, there will be the same cross linkage that will occur. The last 2 here too, the same thing can occur between them. So you will have the cross linkages between these peptides, which are, residues on the N-acetylmuramic acid component of the glycan. And so in the end, you see the enzyme dissociates, leaves. The cross linkage is formed. So when all the cross linkages are formed, you now have that, full structure of the peptidoglycan, which is the cell wall of the bacteria. It is this protein, the glycopeptide transpeptidase, that antibiotics like penicillin target. So if we go ahead, we'll see that you have, so here now we have now introduced the penicillin, looking like 9 or 6 these structures over here. Okay, so you see the enzyme because it's occupied with the inhibitor, that is the penicillin, has gone to bind in there. Now, when you take a look at the structure of penicillin, so here too I have intentionally brought the structure of penicillin and put it side by side against the structure of the D-alanine peptidoglycan ends, the peptidoglycan terminus. What I'm referring to is that you remember that this enzyme, the peptidoglycan transpeptidase, when it binds, so let's look at number 2. Number 2 is showing it well. When it binds, you see that you have alanine, red, which is that uh, peptide residue that has interaction with the active center, the serine residue in there. And you also see the last end of that second peptide from, the second peptidoglycan that comes to bind to that longer part is also ending with two alanine, residues at the end there, which means the enzyme interacts with alanine. But the bond is formed between alanine and lysine. So you see the structure of the D-alanine ends is what we are showing here. The kind of bond, which is, like lysine, looks very much like, penicillin, except that you see penicillin, when it is introduced into the system, first is inactive in this form because you can see that it forms a ring structure. If you take a very close look, you see that it is this particular part that is forming that ring structure there. And then you have this part also open. So all this part is open, but in penicillin, it is cyclic. It's forming a cyclic structure. But once it starts reacting with the enzyme active center, it gets active. That's what is being shown here, that you see that you have a cyclic structure, but it begins to interact with the enzyme active center and opens up. The peptidoglycan, the alanine terminus, which should bind in the penicillin molecule, will no longer, be able to bind in there. It's some form we refer to as suicide inhibitors. So they come in, they are not active, then they begin to interact with the enzyme, then they become active and block the activity of the enzyme. In fact, you see that inhibition, as you see, I was referring to it as the protease, that glycopeptide transpeptidase is a serine protease. So you see the serine residue is what is being shown here, is what is being shown here to be having a hydroxyl group on it. And you have the penicillin coming to bind on that serine residue, to give you that, penicillin have many antibiotics that are having that functional group structure, what we refer to as the beta-lactam antibiotics. They have these types of rings, like you saw in penicillin. You have that square ring with the 5-sided ring fused to it. In some, you have these 6-sided rings. So we have different types of these beta-lactam rings in different types of these antibiotics. Of course, this is not exhaustive. Even if you look at this, you see amoxicillin is having I think the first ring there. So that will be a pinam, because it's the first ring over there that you find inside amoxicillin. But if you take a look at, so these are some, I think, the most common antibiotics you'll be finding around in Ghana. If you see clavulanic acid, for instance, its ring is even not there. That is an oxapenam. So it's a penam, but in place of this sulfur that you find there, in place of that sulfur inside there, you see that you have an oxygen in there. So in clavulanic acid, you have an oxapenam, for instance. Think so you have a combination of these now, on the Ghanaian market, we have this amoxiclav. So it's a combination of amoxicillin and clavulanic acid, to make it to increase efficacy of the activity of the antibiotic against the bacteria infections. All right, we can go on to a different story. This one is, okay, so biosynthesis of, is somebody writing something? Okay, somebody left. Biosynthesis of prostaglandins. Prostaglandins are these biomolecules that are responsible for pain recognition, like in times of inflammation. They sort of cause some form of vasodilation. So it can be flow of blood. Some of them also cause some blood clotting. So opposite ways they can regulate these things, like the thromboxanes, for instance. They go into blood clotting, but things like the prostacyclins will be in the opposite, But actually, what we'll focus on will be just up to, say, synthesis of prostaglandins, from arachidonic acid. And the enzyme which is involved there are some cyclooxygenases. So all of these, fine, you will see that you have different pathways in this process, synthesis of prostaglandins, and they go further to form prostacyclins, thromboxanes, that will go ahead to form, some other forms of thromboxanes will go into different activities. But we won't go into all of this. Like we did for the previous one, we just focused on one reaction, like I say, one arrow at a time. So we just take the one which is relevant to us. So let's go on. So we take the one which is relevant to us. This is the first two reactions, diacylglycerol, phospholipids. I mean, those are, I think when we continue to look at metabolism, and then we get to metabolism of lipids, we'll come to see some reactions like this, where you see diacylglycerol going into the synthesis of arachidonic acid. And this arachidonic acid can go into the synthesis of prostaglandins. What I mentioned, that the prostaglandins will be acting as some pain recognition, biomolecules. And it is actually the synthesis of these prostaglandins that some, these non-steroidal anti-inflammatory drugs target. This time we are looking at a common drug, brufen. Brufen targets, cyclooxygenase 1 and cyclooxygenase 2. Sometimes even some of the drugs, maybe some of you are working in pharmacies, you see some of these drugs, say COX-1, COX-2, things like that. It's referring to those enzymes, cyclooxygenases, that these drugs are inhibiting. So all brufen is doing is to inhibit these cyclooxygenases that are involved in the synthesis of prostaglandins. In that way, pain will be reduced, because prostaglandins, I mentioned, they are biomolecules involved in pain recognition. So if their synthesis is inhibited, then pain is relieved. So that is another example of inhibition being applied. Then we can take say aspirin. Aspirin is also another pain reliever, and you see it's also inhibiting a cyclooxygenase. Same reaction, arachidonic acid getting converted into prostaglandin, and you have aspirin that is inhibiting that. But of course, this aspirin is a special case because it does that by what we refer to as irreversible. It's irreversible inhibition. I think previously what we were looking at for brufen is reversible, but this is irreversible. So similar reaction, aspirin is doing the same as a non-steroidal anti-inflammatory drug. Now, I think it's just okay. What time is it? It's already 30 minutes left. So I think this will be the last one, and then we can open the floor. And I know so many of you are working in maybe pharmacies and things like that. You have more to tell us than I am saying. We have reversible inhibition by diisopropyl fluorophosphate. This time around is in say insecticides that you can have some organo phosphorus pesticides that target acetylcholine esterase, the enzyme. We saw acetylcholine esterase, acting in muscle contraction, muscle as a neurotransmitter, so in the nervous system. And so you see DIFP acting as an inhibitor. Here again, you see the enzyme is having what at the active center, the active site, serine. So that's what is being shown, OH. So you have the side chain CH2OH being shown there. And you have DRFP that comes to bind covalently to the enzyme. So once it binds covalently to the enzyme, which is acetylcholine esterase, it means it prevents the activity of acetylcholine esterase in that process of muscle relaxation, contraction, where it goes into hydrolyze acetylcholine from muscle cells so that the next signal can come for the muscle to contract. So what it means is that DIFP, its inhibition will result into some paralytic reaction, say some paralysis, because the muscle will just be relaxed. So in an insect, if you have this DRFP in an insecticide, the central nervous system will not work. And once there is paralysis, then eventually it will result into the death of that insect. And in fact, some of these insecticides can also have such effects on humans as well when you are exposed to it over a long period of time. Sometimes, because many of these insecticides, these active ingredients, they are lipophilic. They dissolve in lipids. So when humans are exposed to it over a very long period of time, what happens is that today you are young and you are exposed, and these things you may not feel the effect of it today. But what happens is that a lot of times these components go to accumulate in the adipose tissue that's under the skin. There's a lot of fat there, so they accumulate there as if they are not there. Then later, in old age, or maybe in some disease condition that you start to lose weight, when you have the adipose cells breaking down, the fat is breaking down, then you start to have these compounds being released back into the bloodstream and start to cause some complications. That's when some checks will show that you actually having some pesticide intoxication, things like that. It came long time, and perhaps it was chronic and accumulated. So, which is why we should be careful about the use of these chemicals. I'm very much concerned about, how the farmers in Ghana now, indiscriminately use these things. Actually, these things should be used by professionals. But here's the case, it's all over. You have all corners everywhere. You have agrochemical shops. Anybody at all with no chemistry knowledge just goes, buys. And some of them, when even it is, because they can't even read, it's written on it the amount you should take to dilute before you spray. They feel that they want the thing to be very, effective. So they pour more than what should be there and start spraying all over the place. So sometimes you'll be in your house somewhere and somebody is spraying in things, and you can even sense it in your house where you are. You are not the one doing it, but it is something which is, spreading all over, and everybody is inhaling it and all of that. Some of these things, you may take it as maybe sometimes, like if you know some of these things, so a lot of times you are worried so much about how things go as if it is normal, but it's not normal when those things are going on. And you, as an educated person, now you have this knowledge, you can go out there and advocate for better use of some of these things. All right, now the floor is open. Let me listen to other people's voice. Irene, your hand is up, so let me uh, allow your microphone. Irene, your microphone is up. Irene. Irene Apeja Aqua. Oh, okay. You don't have any question, but I can see your hand is up. That's why I'm calling you. All right. Aya Saline. Aya Saline. Saline, your microphone is enabled. Unmute and speak.
Okay, please, talking about the non-competitive inhibited. When we're looking at the graph, I don't know whether at the end of the day, when it slow down the reaction and the product is formed, that the non-competitive also form a product, or it will just the subscript will just the enzyme will just release it to go.
Okay.
Looking at the graph.
Looking at the graph, products will be formed, but the product will be formed over, like the amount which will be formed over a period of time will be less than if you don't have the inhibitor there. So it's not that the product will not be formed. The product is being formed all right, but the enzyme is being inhibited. So its activity is slow. It doesn't mean the reaction is not occurring. It is occurring. The product is forming, but the reaction is slowed down. That's what the inhibitor does. It is the same thing to do, even if you are referring to the competitive inhibition. Product is being formed, but the activity of the enzyme is slowed down. Is it clear?
Okay, sir. It is clear.
All right.
Thank you.
All right. We still have like 19 minutes more. Has anybody written any message? Okay, nobody. All right. Abigail Ayongo. I thought I saw Abigail's hand. Where is she? Those that don't have question, they can put their hands down. Okay, I'm now seeing Abigail here. Abigail, your microphone is allowed. You can speak.
Sir, please, concerning the competitive inhibition, please, when you were saying that the more the substrates, the more when the substrates, the more substrates, the more the molecules inhibitors are increased. So I didn't understand that part a bit.
Okay, what I was saying was that if you have some molecules of inhibitors that are bound to the enzyme and you introduce more substrates into the reaction mixture, as you continue to increase the substrate concentration, the substrate can go to displace the inhibitor. Is that what you mean? We're talking
Yes,
about a
please.
competitive inhibition. Yes. So you have substrate that is binding onto the enzyme. You have an inhibitor that can also bind to that same active center of the enzyme. Now, if it is a reversible inhibitor, as I was saying, if you are introducing the substrate into the reaction mixture and you continue to increase the concentration of the substrate, it will go to displace the competitive inhibitor from the active center. So the reaction will proceed. So you increase concentration of substrates to undo the binding of a reversible competitive inhibitor. That's what I was saying. I hope it's clear now.
Yes, please.
All right. I thought I saw somebody else. Everybody has put their hand down. Or it's the first time he asks a question, his hand is still up. Okay, let me see. Chum Berryman. Chum Berryman, your microphone is allowed. You can unmute yourself and speak. Chum has also put. Chum, do you want to say something? I have enabled your microphone. Unmute yourself and speak.
Sir, please, I want to ask about the irreversible inhibitor. Where the other side of the enzymes, the inhibitors go and bind it, won't it influence the desired end products of the reaction?
Won't it influence what?
The desired end products between the enzyme and its substrate. When the inhibitor that side, won't it influence won't it change the product to something else, or it will just give us the desired product to be formed?
Okay, so if you have an inhibitor which is binding to the enzyme, you are talking about this non-competitive inhibition. When it binds to it,
yeah,
it prevents it prevents the reaction from proceeding. So the reaction will not proceed. The inhibitor goes to prevent it. Is it clear?
All right, sir.
So it's not that the product will be formed by it to be something else. The product will not be formed at all. Like it will go and inhibit that process from proceeding altogether. Okay, we still have like 13 minutes more. It's like many of you are already celebrating Christmas, so they have not come to class. I'm seeing that it's your class, I think, is bigger than this. How many of you are in class? You see, Akpelo Christopher says in your last presentation, will this insecticide get under the skin adipose tissues through contact with a chemical or through inhalation? Okay, I get you. You know, some of these can be through the skin. So as you said, by contact. And some through inhalation, it gets to the bloodstream and it will eventually get stored in the adipose tissue. So it can be both ways. But of course, inhalation, that one, this is that quickly you even start to have some respiratory, discomfort and things like that. So that one quickly, it's noticed that no, you are exposed to something. But it is the ones that can actually, go through the skin that perhaps you may not quickly notice, that you are exposed to such things. So it's not only the skin. It can be skin. It can be just like the way when you take in some like when you breathe in something, it gets into the bloodstream. When it gets into the bloodstream, so many things will happen with it. If it happens that that substance is able to be broken down in the liver as it moves in the bloodstream, you have some detoxification, and so it can get broken down and perhaps excreted through urine out of the system. But imagine that if it goes to form some conjugates, and those conjugates, move into storage, into fats, then that is when it can get dangerous. So most of it, obviously, through the skin, yes, will be the major, way of it getting stored in the adipose tissue through the skin than through the, respiratory system. All right. We still have 10 minutes. I was asking how many of you are in class. Nobody has written anything. Is a class rep here? How many of you are in class? Because it appears you are enjoying Christmas already. 72. Oh, really? You are sure about that? Because I'm seeing that 70, somebody say 73. Oh, okay, then it's like almost all of you are here. It's just, I see. Oh, then you are, let me say you are all here. If anything at all, maybe one or two people are not able to come. I see. That's fine. Emmanuel, okay, maybe Emmanuel is asking you those in the class. Obviously, this question is not to me. He says, where is Shadrach? So whoever knows. Then Shadrach now comes to say you are 82 and counting. And counting. I see. When you are in the middle of the semester. Anyway, this one, the good thing about it is that the lectures are being recorded. So if anyone who is serious, if you sit down and you listen to all the lectures, and make notes, look at the slides, I think you'll be okay. So, but that will mean you will really have to sit down, and if you didn't go through it, step by step like that. There are people who are still getting admission. Okay, that is according to Shadrach. I see. So this is what happens. Shadrach tells me today 82, and then by the end of the semester, when you are going to write exams, and I have more than 100 people. What Christopher says, he thinks that the slides that I sent is finished. Oh, slide. This one we have done now. After the lecture, I will send about the recorded lecture. I'm not able to open the video. How? How are you not able to open the video? Because I always send the link. Doesn't Shadrach Shadrach says he's no longer the class rep. And so those who contacted me the last time, the ones I sent the link to today, they will send you the link. So all this while, Irene has not been able to watch any of the videos. How come? Kenneth Wellington says, please, how does an inhibitor binding affinity to bacterial enzyme compare to its affinity for a human one? That's a very general question. For example, when both the pathogen and the human have similar enzymes, I wish I have such an example to give. You have a pathogen enzyme, and then you have a human enzyme. That is, maybe you are referring to, in fact, in drug discovery, in drug design, you always, will work on the specificity of the active substance that should be binding to the active site of an enzyme, of a pathogen, so that you don't introduce the substance into the system that will go and rather be fighting the host. Of course, you have situations where, like for instance, if you are taking a methotrexate that I referred to, there is biosynthesis of hydrofolic acid also in humans, so that in chemotherapy, where you are using methotrexate that should go and be fighting cancer cells, for instance, they can also have effect on normal cells as well, which is what results into like loss of hair, vomiting. Those are side effects that should be occurring. So even if you have a drug substance which is having some binding onto the host's enzymes as well, some having, that will be what you are referring to is an adverse effect of a drug. Engineering of the drug always, will be to reduce these things, which is why a lot of times there's even improvements upon this. Sometimes we look out for different isomers that will be specific to, the pathogen than the host. Okay, so Irene says, I was able to watch the first video, but not the remaining ones. Wow, that's interesting. How come you couldn't watch the remaining ones? I think there are even, when I end the lecture, yeah, those of you who are still online even before, that's why it's even better that you don't leave the class before I end the lecture. Because when I end the lecture, in the chat, the video actually comes there. So there is an original link which comes on Teams that you can have access to. Before, when I download it myself and upload it, because the upload it onto YouTube. That's the link I give you. That link is there for forever. You can watch it at any time. So you have the video at these two different places. So today, don't leave the class before I end it. The moment I say see you next time, don't leave. I'll just end the meeting. When I end the meeting, you see that in the meeting chat, the video will come there. So you can have the original link from there direct on Teams. Irene, I hope you get it. Okay, so how about, okay, Kenneth, are you okay with your interesting question that you asked that I tried to, answer? I hope you are okay with it. Okay, Kenneth is fine. All right, it was an interesting one. It's 1 minute to time. Okay, Kenneth says he just read that they are mostly concentration dependent. That is, you are referring to the affinity. Concentration dependent, the higher the concentration. So that what you mean is that that issue can be dealt with dosage. That's what you mean. So that's when you have dosage optimized, then you'll be having the activity on the pathogen instead of the host. All right. Some people have already left. A lot of you have already, about more than almost 20 people have already left the class. So I mean, we have come to the end of the class, and I would say till we meet in next year. All the best. Enjoy your holidays, and don't forget to revise biochemistry all the time. Bye for now.